So I really quickly wanted to talk about the different tweets and how you're doing. So one of the things that I'm noticing still for a few people is if the article has been tweeted, so remember to first search Uaguel Food Prof and the hashtag of the first author's last name. If it's there, irrespective of the week it's there, tweet under that one. Okay? So if you're tweeting and someone else has already tweeted on the article, you're going to reply to that tweet as opposed to starting a new thread. So most people are doing it. Yes? It doesn't matter what week it is. So it doesn't matter what week it is. So that's the downfall to using a really old paper. So if you tweet about a paper in the first week, it's going to be buried in my thread. Yeah. But you'll still get activity through yours. Okay. So now we're moving into the stuff that I know more about. So we're moving out of the basic nutrition and we're going to start moving into different aspects of food science. And today what we're going to start with is really what underlines, why is none of this working? There we go. My clicker's not working today, so I have to be locked behind this table. So today we're going to talk about food safety. And most of what we do to foods is to ensure that we don't have an outbreak associated with our product. So a lot of the formulation, meaning we add sugars, we add salts, we add preservatives. So it's not only the chemistry that we're doing to affect the food safety, but it's also the processing. So why do we pasteurize foods? Why do we heat foods? Why do we use other non-thermal strategies to try to slow down the growth of food? So when we talk about food microbiology today, our food industry has changed radically in the last 100 years. So when we talk about production of commodities or production of different products, we have to remember now that we work in mass farms, in mass farming. So what that means now is that instead of having isolated outbreaks that may affect a few animals or a few products, we now have massive recalls associated with outbreaks. So this is in part due to the fact that we have large amounts of animals living in very close proximity. So we can talk about herds of cattle, pigs, or chickens. But also remember that we have very few slaughterhouses. So what happens is when we have an animal product going to get slaughtered or an agricultural commodity that's going to get processed, these farms all send their product to a very few processing facilities. So when we have an outbreak associated with a processing facility, this may be producing thousands and thousands of kilograms of food a day. So now when we have a recall associated with these or an outbreak associated with these mass-produced facilities, we are far more likely to get a lot of infections associated with that. So now instead of having a lot of isolated incidences, which is what it was 100 years ago, what's happening today is that we get very few outbreaks, but when we have the outbreak, many, many, many people are affected with that associated causative agent. So again, this is because we have mass production facilities both from the farm production but also as a processing facility, and we centrally process those foods. So in this area, we have one or two slaughterhouses. We have one mainly for pork and one mainly for beef. So all the cattle come into this area to get processed. So again, if we have a recall associated with that processing plant, which we'll talk about today, Nawaz, can you grab some paper towel? We have a spill. It's okay. So when we have a recall associated with one of these processing plants, again, it affects thousands and thousands of kilograms of product. So when we talk about the purpose of food processing, depending on the product we're working with, we have different outcomes we're hoping for. So first, of course, irrespective of the process we do, we always want to increase the safety of the food. Now, when we increase the safety of the food, we are trying to eliminate food hazards. And when we talk about hazards, it can be anything from pesticides to antimicrobial residues. And then the one we think of most often is the microbiome, or the microbiota that may affect it. So how do we eliminate pathogens if they're present, and how do we prevent pathogens if they're not present but being added after the fact we process them? So once you've opened the packaging, or once you've started to consume that food, how do we prevent recontamination? Now, when we do this, all of these processes require some kind of disruptive energy that you're putting in. It can be heat, it can be irradiation, it can be pressure. These not only affect the microorganisms that are present in the food, but they also affect the quality of the nutrients that are there. So we'll talk a lot on Thursday about nutrient degradation during thermal processing. So anytime you cook a food, if you boil your food, if you cook it in an oven, irrespective of what you do to it, you are damaging the nutritional quality of that food. So again, every time you cook a meal, you are reducing the quality or the amount of nutrients that are present. So from a food processing perspective, we are very, very conscientious of this. And we talk about already the difference between raw milk and pasteurized milk. So if you consume raw milk, the milk is a lot more white in appearance. So if you drink pasteurized milk, it's got that slight brown color. Again, anyone remember what reaction leads to that? Maillard reaction. Again, Maillard reaction is a ubiquitous reaction whenever there is a reducing sugar and a protein and heat being added. Now in the carbohydrate section, we'll talk about how different sugars accelerate that reaction comparatively to other sugars. The other thing we try to do when we process foods is we try to increase the convenience of them. So think about a can of soup. The purpose of that is to ensure that you don't have to actually go and make that soup. So we're trying to get convenience in there. And then favorable economics. And this is a big one. This is something we take for granted in Western society, is that we process foods most of the time to ensure that we don't have losses. So we can take, again, corn. So in Canada, we have about 5,000 farms that produce enough corn to meet the sweet corn market. So the sweet corn market is corn on the cob, canned corn, or frozen corn. The other 50 or 1,000 so farms associated with corn is to make commodities and to make ingredients. Cornstarch, corn syrup, high fructose corn syrup. All of these different commodities allow us to take corn, which is perishable, extract the ingredients we want, make functional ingredients so we can design foods that have a very, very novel or useful structure, but we can also preserve that food. So then we have cornstarch, which once it's extracted from corn, has a shelf life of years. So again, we're taking something that's highly perishable and we're making it available year-round or seasonally. Now, when we talk about causes of deterioration, there's a lot of mechanisms to deteriorate foods. The ones we most commonly think of are things like pests. So again, when we talk about corn production or agricultural commodities that are being growing on a farm, they're exposed to insects, rodents. In developing countries, this becomes much, much more of a problem. We also have microorganisms. So we can have spoilage due to bacteria, we can have spoilage due to yeast or molds. We also have chemicals that can be in our foods, things like pesticides. We can also have biochemical reactions. So when we harvest that commodity, that product, that corn, that wheat, whatever it is still undergoing respiration. So it's undergoing metabolic pathways which can be detrimental to that food. So things like oxidation, enzymatic browning. We'll talk about these later. And there's physical factors that affect the stability of our products. Light, exposure to UV energy, oxygen, so oxidation of oils. So I always like to ask the question, how many people here use what I call a novel oil for cooking? So at home, does anyone use something like safflower, sunflower, flaxseed oil, fish oil? Who uses those regularly, or who has a bottle of them in their kitchen? So about 20 people, so about 10% of the class. Where do you guys store it? Where do you keep yours? Cupboard? Cupboard? Where else? Cupboard? Does anybody restore it in the fridge? One person? So the rest of you are actually exposing yourself to carcinogens, because oils very readily, especially the ones with flaxseed oil, fish oil, these are extremely prone to oxidation. So if they're at high temperatures and room temperature is high enough, and if they're stored in the presence of light, you're going to undergo autooxidation. So if you don't use your olive oil or fish oil quickly, you'll notice off flavors starting to get associated with them. You'll start to smell that rancid type, it's kind of like a rancid foot smell. So all of these things we have to process foods for in an attempt to increase the shelf life. Again, this is so we can have commodities available year-round. So what do we need to take into account? We need to obviously decrease the rate of microbial growth, right? We know that pathogens and spoilage microorganisms are ubiquitously found in the environment. So something like Clostridium botulinum, one of the most toxic neurotoxins known to man. Also, this bacteria is ubiquitously found in the environment, meaning that when you harvest a crop, it has Clostridium botulinum present. So when we process foods or when we harvest foods, we have to ensure that we eliminate any microorganisms that may be pathogenic. Obviously, we don't want a foodborne outbreak associated with it. We also try to reduce the amount of spoilage microorganisms. We want to have a longer shelf life. There's different ways we can modify that food in an attempt to limit the microorganisms that are growing. So what do we do to foods? We can add preservatives, right? We can add salt and sugar. We can add nitrates, phosphates, sulfates. We can add other essential oils, things like rosmarinic oil. We can add different fermentation products. We can add ethanol, which kills microorganisms. We can add lactic acid. So we need to destroy and inactivate all microorganisms associated with pathogenesis when we talk about food products. So how do we typically do this? We do it by heating that food. And again, we've already talked about why that's detrimental. We have sensory properties that are changing, right? We have the Maillard reaction, so the visual appearance, as well as off flavors being produced. We have degradation of nutrients. The longer you hold that food for the temperature, and the higher that temperature is, the faster we reduce the quality of that product. So now, when we process foods, we want to minimally process them and have them still be free of any pathogens. So we heat to extremely high temperatures for extremely short periods of time. So milk will heat to 72 degrees Celsius and we'll hold it for about 15 seconds. If we heat it to 62 degrees Celsius or 63 degrees Celsius, that same amount of sterility, we then have to hold it for 15 minutes. So we can play, and we'll talk about this on Thursday, we can play with the time and temperature combinations to try to preserve the nutrient quality while still reducing the population of microorganisms. The other thing we need to do is restrict access to microorganisms during the food's life in the supply chain. So we do this by putting impermeable packaging in it. We create hermetic seals in cans or seals in aseptic packaging that are impermeable to microorganisms. We also ferment foods to increase the likelihood of preservation. So why does fermentation preserve different commodities? Well, one reason is, in the alcohol industry, is that ethanol is a very, very good antimicrobial. So when your product has a little bit of ethanol in it, it is going to have a very, very long shelf life. So this is beer, wine, and any spirits you drink. The other thing that happens, though, especially in the fermentation of dairy products, is the formation of organic acids, things like propanoic acid, acetic acid, and these have an antimicrobial effect as well. We also produce other small molecules, things like diacetyl, carbon dioxide, ethanol. All of these things will impede microbial growth. So we can use fermentation as a way to slow microbial growth of pathogens. And this has become a huge industry in the last few years. So we have things like buttermilk. Now, buttermilk's a little bit of a weird one because depending on your source of buttermilk, it may or may not be fermented. But we have things like yogurt. We have bifidus milk. We have some other ones like kefir that are not very popular yet in Canada, but internationally are very, very important. So these don't rely on the production of ethanol, right? there's no ethanol in your yogurt. If you eat a whole bunch of yogurt, you're not going to go out to Trappers or Palace afterwards. You will, but you won't have as much fun. So when we talk about yogurt, and yogurt is a really interesting one because yogurt relies on two cultures. So if you remember when we were talking about the cheese, or the cheese video, if you haven't watched it, we added a single culture. In yogurt, you always add two different cultures. You add one with lactobacillus, and you add one with streptococcus. Now, what's really interesting about yogurt is that these two microorganisms compete for the food source, for the glucose, for the galactose. So they're metabolizing those products and they're creating metabolites. Now those metabolites can have synergistic effects. So those two bacteria can cooperate with each other, meaning a product from lactic acid bacteria can stimulate the growth of streptococcus. And the rate of growth is dependent on the optimal temperatures of those microorganisms, which is not the same. So typically when we talk about yogurt fermentation, we have to hold that culture at 43 degrees Celsius. If you change that ratio or that temperature, it's going to change the ratio at which those microorganisms are growing. So we'll talk about why that's important. So for example, if we start with our milk or a dairy product, we have lactose, and that's what the microorganisms are primarily using for an energy source. So they can split that lactose into glucose and galactose, and the lactobacillus can convert the glucose into lactic acid. So can the streptococcus. Now the galactose will be converted by lactobacillus also to lactic acid, but the streptococcus cannot interact with the galactose. So it doesn't ferment the lactose. So if you have a temperature or have not added sufficient streptococcus, we're going to overproduce lactic acid because the galactose cannot be metabolized by the streptococcus. So we get lactic acid and then galactose. If we take glucose, it can go through the basic metabolic pathway. It can produce pyruvate and acetaldehyde. Acetaldehyde gives it that nice buttery taste. And then that can be converted to diacetyl. Diacetyl is a compound that we often add as an artificial flavor to things like popcorn. Gives it that buttery taste. Now, threonine, which is just an amino acid, can be converted by lactobacillus to glycine. Lactobacillus. The glycine then goes up and stimulates the production of streptococcus. So this shows a synergism between the two bacterial species. So again, that temperature and that relative ratio of those two microorganisms plays a very, very big role in the tartness of your yogurt, how sour it is. It also gives it, you can also tailor that buttery or that diacetyl flavor. The other thing that happens is once we get the increase in pyruvate, that can then begin to produce more carbon dioxide. That carbon dioxide can then go back and stimulate the growth of lactobacillus. So again, these bacteria are very synergistic. Now, streptococcus also produces two other flavor compounds, formate and acetate. Glucose can also be converted to the oleosaccharides. Oleosaccharides are why the viscosity of yogurt is so high compared to milk. So it's these long chain polysaccharides that are being produced. So in yogurt, we have a lot of lactic acid. And this lactic acid is what gives us our preservation. So we often add, or as a byproduct of fermentation, produce lactic acid, acetic acid, and propanoic acid. Now these three acids specifically are very, very important for food preservation. This is why all fermented products have a shelf life of months compared to milk, which is weeks. So it also provides some of the flavor, the mouthfeel, but most importantly is it extends the shelf life. So at very, very low concentrations, again 1 or 2% lactic acid, they can prevent spoilage. They can prevent pathogenic microorganisms from colonizing in the food and actually producing foodborne illness. And it's effective against all microorganisms with the exception of viruses. Now, why does lactic acid become an effective way to impede microbial growth? Well, there's two different aspects to inhibiting microbial growth. The first is we can prevent the growth of microorganisms in food. So that's referred to as bacteriostatic. So bacteriostatic means that the compound is in high enough concentrations that the microorganisms can't replicate, but in that environment, those microorganisms are still viable. So when they go into a different environment, they can then cause a host of problems. If we get above 2%, we become bactericidal. And what that means is the bacteria will actually die in the presence of that organic acid. Why are the organic acids we're talking about C2, C3, and C1? We're not talking about anything longer than things like butyric acid. So very, very short chains. So this works because in most food products, our pH is at a point where that acid is protonated, right? So remember organic acids. So we have some carbon chain, doesn't matter what length that chain is, with a carboxylic acid group. Now, every acid has a dissociation constant or a pKa associated with it, right? Now, everyone remembers pKa's from first year chem, hopefully? So, if you are above the pKa, the dissociated state will be different than if you're below the pKa. So the pKa really measures at what pH that hydroxyl group will leave. Now when we talk about organic acids, the pKa of these are typically fairly low, 3.8 to about 5. Most foods we consume are above a pH of 3.8, meaning they're mainly associated. The hydrogen is still on that carboxylic acid group. Once we drop below that point, so again, we're protonated. Once it moves into the cytoplasm, it changes the pH of the cytoplasm. The cytoplasm is closer to 7. This allows then a dissociation of that organic acid. So what does that mean? So associated organic acids that have their hydrogen are able to pass through the cell membrane of that microorganism. Once they move from the food to the cytoplasm, the pH changes, the amount of associated and dissociated acid changes, and by doing so, what happens? What happens when we get the accumulation of hydrogen within a cell? The pH drops. What happens when the pH drops? What happens in our stomach? Proteins begin to denature, right. Now when those proteins are denaturing, in a microorganism, what are proteins used for? They're the hormones. They're the signaling molecules. They're the enzymes. So by dropping the pH of that microorganism cytoplasm, we denature all of its enzymes. If we denature enough of those enzymes, we kill the microorganism. So when we talk about organic acids, why they work comparatively to the long chain fatty acids are not as readily able to cross that cytoplasm. So we can't get enough long chain fatty acids into that cytoplasm, so they don't dissociate. Now, this only works if the pKa of food and the bug is similar. Once it becomes too dissociated, the microorganism can prevent the uptake of hydrogen in its environment. So only associated acids can cross the cellular membrane. Now, when we look at this example for yogurt, has a pH of about 4. Lactic acid has a pKa of about 3.8, so we're pretty close to the pKa, meaning that half of that acid is essentially associated and half of that acid is dissociated. The half of the acid that is dissociated, meaning that it's lost its hydrogen, is unable to cross that cellular membrane. So it doesn't play a role in decreasing the pH of the cytoplasm. So what happens? Well, when the associated organic acid crosses into the cytoplasm, the pH goes up. So our dissociation constant, we are moving further away. So what happens? The organic acid that was associated becomes dissociated. So now that hydrogen is released. Now microorganisms are very sensitive to pH. They have a lot of mechanisms to pump out that excess hydrogen. So if we have a concentration of about 1%, that microorganism is spending all of its energy moving that hydrogen out. So it's unable to replicate, and because it's unable to replicate, because all of its energy is being used elsewhere, it doesn't grow in foods. Now, if we get up to 2%, that microorganism doesn't have enough proton pumps to get rid of that dissociated acid. The pH begins to drop. That drop in pH is associated with a denaturation of the enzymes. That denaturation of the enzymes ceases its metabolic pathways, and the microorganism dies. So when we get enough lactic acid or organic acid in our food, we can actually begin to dissociate only the microorganism. We can dissociate that acid once it goes into that microorganism. Now there's also organic acids within our food supply. So certain fruits, certain vegetables have organic acids, things like rosemary. And a lot of these novel extracts that have essential oils are starting to be looked at for their antimicrobial properties. So when you look at things like sausages, you'll notice they put things like rosemary extracts. And the reason they're doing that is they want to incorporate those essential oils, again, that have an antimicrobial effect, to try to eliminate the need for other things we add to cured meats. Things like nitrates. Nitrates are really, really good at preventing microbial growth. So if we remove nitrates and do nothing to those products, we're going to get spoilage. We're going to get microbial growth. If we add enough essential oil, we can in fact start to preserve that sausage without the need then for nitrates. The other thing that's really important is that we prevent spoilage. So a lot of what we do in the food industry really tries to understand how can we increase the shelf life of a food product. And again, when we talk about spoilage microorganisms, they're not going to be a cause of disease, but they're going to be a huge problem when we talk about food loss. So here's a little fun thing we're going to do. So pathogenesis is associated with disease. So this is things like Salmonella, E. coli, Clostridium, norovirus. Now the estimates are about 128,000 people a year are hospitalized, and about 3,000 in the US die per year. Now out of curiosity, so here's the first Twitter poll. What foods do you think are most likely to be associated with a foodborne outbreak? Have you sent it yet, Nawaz? So let's see what we think it's going to be. So you have to click on the See New Tweets at the top. That should put in, oh, there you go. And then just continually hit refresh. So we've got produce. Where'd that go? Dairy and eggs. Only 2 votes. Come on. Here we go. People are going. Yes?
Does it matter like how you process it though? Like whether you eat produce raw or...
Absolutely. So we'll talk a little bit about this. But the question's asking, what do you think are the most likely causes? So this is associated with just foodborne disease. So we're up to 56 votes. Once we get 100, I'll tell you what the answer is. just click here and hit enter. So dairy and eggs have dropped. Produce is still low. All right, so the answer is actually produce. Produce is the most likely cause of foodborne pathogenesis in North America. And we'll talk about that in a minute. Now I want to know, how many of you have had a foodborne illness? So 73% say yes, 27% only say yes, and 73% say no. Wow. Now the next question is, have you ever been diagnosed with a foodborne illness? So not have you think you've got one, but has the doctor ever confirmed that you have Salmonella or E. coli? And this is one of the challenges right now in surveillance of foodborne outbreaks, is that hopefully this number drops. So now we're down to about 7%. That reflects, again, so when we talk about 128,000 cases, just based on our numbers, we have about a 3-fold underreporting, which is about what the industry norm is. So when we report cases of foodborne outbreaks, we are reporting confirmed cases of foodborne illness. So if you throw up for a day or if you get diarrhea, chances are you're not going to run to the doctor. You're going to wait a week or two. So we underreport the cases of pathogenesis always. And this is something we always have to consider. So again, when we talk about the risks associated with production of food, right now in North America, produce is the leading cause of outbreaks in foods. And we'll talk about why that is in a little bit and some of the challenges that are associated with it. This is typically followed by meat and dairy pretty evenly, and then fish and shellfish. So about 1 in 8 Canadians per year are affected with a foodborne illness. There's about 230 deaths per year and about 11,000 hospitalizations. Again, these are only confirmed cases. So when we talk about the major foodborne outbreaks in Canada, this is norovirus. So norovirus you always hear about associated with cruise ships. So when there's a big outbreak on a cruise ship, typically it's norovirus. Listeria, another big one. Anyone remember the big Canadian outbreak of Listeria? You guys are all old enough that you should know it. Anyone remember the company? Maple Leaf Foods had a massive Listeria recall in 2008. We've got Salmonella, which again, there's a current outbreak going on now that they're trying to contain. E. coli 0157, another really important Canadian outbreak. What was E. coli 0157? Walkerton, the water supply in Walkerton. And then we have Campylobacter. So these are the big 5 in North America and in Canada. Now, when we talk about different hazards, is any foodborne illness is any illness that is caused by the ingestion of food. Most typically we think of the incorporation of biological hazards, microorganisms, bacteria, yeasts, molds. When we talk about chemical hazards, these are fairly rare. There was one a few years ago in China where they were adding melamine to milk. So why, does anyone know why they added melamine to milk? What was the purpose of that? Yeah. Yes, and do you know why they wanted to increase the nitrogen content?
Make it look like there's more protein.
Exactly. And do you know why they wanted to make it seem like there's more protein?
To get more sales.
To get, kind of. So what they wanted to do was they wanted to dilute the milk. So you add a whole bunch of water, and instead of selling milk, you're selling water at a much, much higher price. The problem is, when you do that, you dilute everything else that's in there, the fat, the protein, whatever else is in there. So what this company did is they took melamine, which has a whole bunch of nitrogen, and it's a very small molecule. So when they did the analysis, the protein content seemed like it hadn't been diluted. So then the the farmers ended up getting paid more. The problem with that is when we talk about things like cheese making, what does that rely on? The protein structure. So if you've diluted the milk and there's less protein in it and you go to make cheese, you've got a big problem. So that milk that was diluted never made it into the cheese industry, never made it into the ice cream industry, because the functionality of those ingredients in milk are required to make good products. So they didn't sell that milk to ice cream, to things like fermented yogurt, because all of those things require that starting material have appropriate concentrations of proteins and fat. So where did that milk end up going? Yeah, infant formulas. And what happens with melamine? Melamine crystallizes in the kidneys. So what happened was all of these babies started to develop basically really horrible kidney stones. And it to the point where there was quite a few deaths associated with that melamine outbreak. Does anyone know what happened to the president of that company? He was executed. So he got the death penalty. There was actually quite a few people associated with that outbreak that were executed. So when we talk about microorganisms in food, there's a lot of debate right now at what level of microorganism is safe to consume. So is it safe to consume 1 or 2 bacterial cells that are Listeria, that are E. coli? So we need to know what the population of those microorganisms are. Microorganisms typically double if they're in ideal conditions every 20 minutes. So that means at 10 minutes, if you have 1 cell, that becomes 8 cells. If we have 2 hours later, it's 500 cells. 4 hours later, it's 2 to the 10 to the 6 cells. And 3 hours later, it's 1 times 10 to the 9 cells. So even though our bacterial populations may initially be very low, if we do not create an environment where they are inhibited, they will grow extremely rapidly. Some of the challenges associated with that are if that microorganism is growing in its ideal conditions, and we'll give Clostridium botulinum as an example, that bacteria does not make you sick. Botulism, the bug itself, isn't that bad. It doesn't cause an infection. What does it do? It produces a neurotoxin. And that neurotoxin is produced when it goes from a spore, which would be when it's in an aerobic environment. When it goes into an anaerobic environment, it goes to an active cell. And that conversion from the spore to the active cell releases the neurotoxin. So if you've had microbial growth in a can, you can later kill that population, but that toxin has been produced. And that toxin is now in that can of soup. So even if that bacteria is dead, if it has had time to replicate and that toxin has been developed, you will get sick from consuming that canned product, even if there's no viable cells left that are still growing when you consume it. So we have to, very early on in the process, try to eliminate pathogens. And we do this by heating foods. So we can heat foods either uniformly, which is novel irradiation techniques, microwave radiation. We'll talk about a few of the different techniques. Or we can heat that food non-uniformly. Traditional thermal processing, so pasteurization and canning. And we'll talk about some of the details about that next week. The food has a set of intrinsic characteristics. And those intrinsic characteristics are things like, is that food exposed to oxygen? If it is, aerobic bacteria are capable of growing in it. So think about if you go to the produce, anywhere in the produce aisle you've got your produce exposed to oxygen, so you don't have to worry about anaerobic bacteria that are on that produce, right? Because they're not going to grow. You have to remove the oxygen for them to be able to grow. So the intrinsic factors determine which bugs are capable of growing in that environment. So the big things we look at are water activity, So how much sugar and salt are added? pH. So there's a magical pH of foods. Anyone know what the magical pH of foods is? Has anyone ever canned food at home? You should know what the magical pH is. Yes, but there's a number. No? No? Anybody? So never, ever, 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 ever can food at home if you don't know the pH. pH of 4.3 is the magic number. And does anyone know why that number is magic? It is the delineation of whether or not Clostridium botulinum will grow in your food. So the intrinsic factors determine the constituents of the food or the environment of the food that will play a role in determining the microbial growth that happens. Does that make sense? So again, if you have a high pH food, more microorganisms are going to be capable of growing in it. If you have an aerobic environment versus an anaerobic environment, this is going to change the population of microorganisms that we have to worry about. So when we talk about bacteria that are capable of growing in an anaerobic environment, so this is things like canned foods, right? Canned foods require higher thermal processes if the pH is above 4.3. So if the pH is above 4.3, we have to heat it as though we assume there's Clostridium botulinum in there. Spores are harder to kill. They're harder to destroy. So we have to heat them for longer periods of time. If that pH is below 4.3, we can do a far less severe thermal process because we know that Clostridium botulinum can't grow. So foods where this is really complicated especially, and I would always recommend never home canning anything with tomatoes. And the reason for that is tomato pH, the pH of tomatoes depends on three things. One is the type of tomato you're using. One is how ripe that tomato is. And also how much water that tomato has grown with. So by chopping up or creating a salsa or a tomato sauce, if you don't measure the pH, you are literally putting your family's life at risk because you could have the growth of microorganisms associated with botulism. So what do you do typically when you can tomatoes? You add some vinegar, right? You want to make sure you add something acidic to ensure that you're below that pH of 4.3. So the only place we really see botulism outbreaks anymore is in very isolated cases where someone has done home canning. We don't see it on an industrial scale anymore. We've been pretty much able to eliminate it. So we've talked about the importance of pH. We've talked about the importance of oxygen. The other one is the water activity. And the water activity is how we get stability in most processed foods. So we want to have a water activity that's, when we're below 0.9, we don't see very much bacterial growth. So the likelihood of getting a pathogen drops significantly when we're below 0.9. When we're below 0.7, that's when we stop seeing spoilage microorganisms being able to grow. So we've talked about pH, we've talked about oxygen, we've talked about water activity. Now we can design foods with what is called hurdle technology. And this is probably the most common preservation technique in the food industry. Hurdle technology uses several sublethal barriers to growth. So imagine so we have our food, we have our bug, whatever microorganism that is. It's going to have a set of intrinsic characteristics that it requires to be able to grow in that environment. So let's say the first thing we do to that food is can it. So we put it in a can. So right away, by canning it, we've converted that environment from an aerobic environment with the presence of oxygen to an anaerobic environment. Now all of the aerobic bacteria can't grow. They don't make it over that hurdle. But all of the anaerobic make it through. So they can still grow, they can still cause disease. Then we change the pH. So we create a second hurdle. And let's say we go below the pH of 4.3. Now, only the bugs that are capable of growing in high acidic environments are going to replicate in that food product. Now let's say we add an organic acid. So we add, let's say, lactic acid. Now again, we've created another hurdle. So only some bacteria are capable of growing in that environment. And by putting enough of these hurdles, eventually no bacteria can grow. So there's none left that are capable of growing. So once we put enough hurdles in place, that bacteria is so stressed that it no longer has the set of characteristics it needs to undergo proliferation, multiply, and then cause disease. So this hurdle technology is used in a whole set of foods called intermediate moisture foods. And intermediate moisture foods are things like cookies, crackers, potato chips, breads, cakes. So when we engineer those foods, we put them and we design them to have a water activity that's low. Again, we want that hurdle in place. Now the challenge in the food industry is, again, imagine a cake with an icing on top. Now that icing and that cake is going to have different water content, right? That's going to equilibrate in time. So the moisture from the icing is going to migrate into that cake. And that water activity of that cake is going to change. So even though we may have had a very stable product initially, it may, the migration of water may change the ability of that microorganism to actually continue to go. Now, when we talk about this, obviously we have to design those hurdles to be appropriate to the food we're talking about. So you can't take produce and add a whole bunch of sugar to it. So it becomes very difficult to actually be able to slow the microbial growth. Now, when we talk about processed foods, what do we like to add to foods? Two different ingredients: sugar and salt. The reason we like to do that is because they bind water. They bind water extremely well. And when they bind that water, they prevent microbial growth. So we can make salt replacers, we can make artificial sweeteners, no problem. We can mimic the sensory properties. There's also ingredients that we can add to get glasses that we'll talk about later, and hard structures. The thing we can't replace is that antimicrobial activity. So even though we may pull sugar out of a food, what then happens to the shelf life of it? We have to come up with another technology, which there's none in existence right now, which is why sugar and salt is found in all processed foods. We have no solution. So when we talk about, again, that limit of about 0.9, things like fresh meats, bread, will undergo spoilage very quickly. When we begin to remove moisture content, things like drying ham, curing meat, fermenting dairy products, things like cheese, maple syrup, increasing sugar content, again, some of these still have high enough water contents that they're able to support microbial growth. As we get drier and drier, the likelihood of contamination drops. So once we get below about 0.85, we don't see much risk of bacteria. We still have mold and yeast that can grow. So to prevent molds and yeast, we've got to get below about 0.6 to 0.7. And then we have a shelf-stable food product. So again, when we design foods, when we process foods, we want to take all of these different factors into account. There's also extrinsic factors. Again, these are environmental factors that control the rate of growth. Again, presence or absence of absence of oxygen. So again, is it canned? Is it in an open environment? Also, what temperature does that bacteria grow at? So no bacteria can grow in the freezer. So below -5, once there's ice formation, we get very, very little microbial growth. But when we're in a refrigerator, we can still see microbial growth. And an excellent example of this is your cold cuts. So if you have things like salami or turkey, if you ever go into your fridge and you take that cold cut you haven't eaten in a while, and there's that nice iridescence on it, has anyone ever noticed that? That's Clostridium perfringens growing. So you've actually got a bug that's growing on there that's causing spoilage. Now, Clostridium perfringens doesn't always cause pathogenesis. It can actually cause sepsis, but it's not typical. So it's still somewhat safe to eat. The problem is if spoilage microorganisms are growing, what else could be growing? Pathogens. So typically when you see signs of spoilage, you're far safer off just to throw it out. So if you see that nice kind of blue and green iridescence on the surface of your salami or your turkey, just throw it out. Chances are you've had some temperature abuse to that product, or your fridge hasn't been able to limit the microbial growth. So when we talk about microorganisms, we separate them into 3 main classes. Psychotropes, which are capable of growing in the fridge. Most psychotropic bacteria are associated with spoilage and not pathogenesis. It's the mesophiles that we really, really have to worry about. The mesophilic microorganisms, their optimal growth range is typically anywhere between 30 and 37 degrees Celsius. And the thermophilic bacteria grow between 50 and 80. These typically, again, are more associated with spoilage, but they play less of a role in food safety and food spoilage. Now, microorganisms can either be strictly aerobic or obligate aerobes, which require oxygen to be present in that atmosphere at 21%. Obligate anaerobes are things like Salmonella, Listeria. Facultative anaerobes can either grow in an aerobic, facultative anaerobes can grow in an oxygen environment less than 21%. We typically in the food industry delineate between aerobic and strictly anaerobic. Anaerobic microorganisms, again, grow in the absence of oxygen. Whenever we talk about a packaging material that is impermeable to oxygen, it plays a significant role. The other thing when we talk about preservation is we often modify the atmosphere of that packaging. So we talk about modified atmosphere packaging all the time. We typically do that to displace the oxygen that is in that environment. We displace it with either nitrogen or carbon dioxide. And again, we do that to prevent the growth of obligate aerobes in that food. But that changes the extrinsic factors, which then allows a different microbial species to grow and to proliferate into that food. So then whatever hurdles we put into place have to be able to accommodate that species potentially being present. So again, we change the pH, we add salt, we add sugar, whatever. Now, when we talk about pathogens in foods, there are 2 different types of pathogens, gram-negative and gram-positive. The difference being if there's a lipopolysaccharide on the surface of that microorganism. You should have talked about gram-negative positive bacteria in first-year biology, yes? Everyone knows the difference between a gram-negative. You should have all done a gram stain, right? In lab, no? First-year biology? They don't do gram staining? Wow, that is a shame. Yeah.
I'm in microbiome now and I'm just learning this.
Well, what year are you in?
Second year.
Wow. The failure of the education system. I was taught this in high school. So, why is this cool? Well, the presence of lipopolysaccharides are what induce fever. So if you have a fever associated with your foodborne illness, you can then classify that as being a gram-negative system. So when these LPS, or these lipopolysaccharides, go into the body, they produce this fever that is associated with an elevated temperature. So you can actually narrow down your bacteria that's present. And there's a professor at Guelph who is a Tier 1 Canada Research Chair, which is the top of the food chain at Guelph. And he does a lot of really interesting stuff using awesome, really high-end microscopy. So SEM, scanning electron microscopy, where you fire electrons and then look at how those electrons bounce off, and you can draw a picture of that food based on this. You can see down into nanoscale resolution. And by doing these techniques, he's been able to look at how these antigens are able to reside at the surface. So when we talk about pathogenesis, there are three main types of diseases caused by microorganisms. There's foodborne infections. Foodborne infections require that bacteria to be consumed living. That bacteria then comes in and colonizes your intestinal tract and begins to produce negative effects associated with the colonization. So what happens is the bacteria can then interact with the villi and start to produce different compounds. This can cause a lysis, or a destruction of those epithelial cells. This can cause, first, a watery stool, so diarrhea. The second thing that this can cause is blood in your feces. So typically when you see an infection, this will associate with the microorganisms actually colonizing the epithelial cells. Make sense? Again, the incorporation of these bacteria can lead to things like vomiting. If they're gram-negative, you can get a pyrogenic or a fever, a pyrogenic effect or a fever. You can also typically have longer onset times. Again, because it needs time, that microorganism, to actually colonize the GI tract. So typically, if you get a foodborne infection, you don't get the symptoms until about 12 hours later. This makes identification of bacteria that are associated with disease very, very difficult to identify, because now sometimes you're talking 2 or 3 days later you start to feel the effects. So you don't necessarily associate which meal you had that caused that effect. So salmonella, the onset depends on the person, how good their immune system is, and again, it can vary between 12 and 36 hours. Now, with salmonella, again, it's gram-negative, it's got the pyrogenic effect, so it produces fever. Typically associated with salmonella is also diarrhea. Salmonella is extremely commonly found in poultry. There is no time you should ever eat a turkey tartare. Turkey is always contaminated with salmonella, so you have to ensure that you put an adequate thermal process into that turkey or chicken. Now, Mary Malone. Has anyone ever heard of Mary Malone? One person? 2 people? What was Mary Malone famous for? Yeah, so in salmonella, you can be what's called a carrier. So salmonella colonizes your GI tract, but you don't have any symptoms. Now, with Mary Malone, what was really cool is she had an infection, I believe, in her, either in her gallbladder or her appendix, I forget which. And it was there for years and years and years, but she was asymptomatic. She never had any effect. So she was living in New York in the early 1900s, and she worked as a cook. She had the first infection, or the first outbreak associated with her, caused 47 illnesses, and 3 people died. Now, they ended up figuring out who she was, and they quarantined her. And if you ever go to New York City, if you go and you see the woman holding the Statue of Liberty, it was gone. There's 2 little islands just off there, and they were actually a quarantine facility for the US for years. The reason they had it there was this is where most of the European migrants came through. They would come through Cook Island, and they would come through this area, and if they were sick, they would go directly into quarantine. But they also used it to quarantine people with tuberculosis, and in this case, Mary Malone, who had salmonella, and she was quarantined for 3 years there, not allowed to leave, in a really restricted facility. Once she got out, she changed her name. So she ended up coming back out, and she got a job as a launder, like someone that does laundry. But the problem was she wasn't making enough money. So she changed her name and went back into the food industry. And again, there was another massive outbreak associated with her, and she was quarantined for life and died in quarantine. And it's funny because they actually offered her because they identified where the I forget if it was the gallbladder or if it was the appendix, but they said we can do this surgery and remove it, and then you can go back out and do whatever you want. She refused the surgery and had to stay in quarantine for the rest of her life. Does anyone else know who this person is? There's another nickname she has that I bet will make a lot more people know who she is. Typhoid Mary. So if you're a comic book geek, Typhoid Mary has been in all kinds of comic books. Things like Daredevil. She's been in a lot of the, what's the other comic series? Oh, it's gone. It's out of my head. All right, Listeria. So Salmonella, we've talked about now. Again, gram-negative, causes a fever. Long onsets, again, a foodborne infection where the bacteria are what actually cause the disease. Now, Listeria is another really, really interesting one. Again, it's a foodborne infection. Do you have a question? No, just waving? Are you making fun of my hand movements?
No, we're trying to figure out which lecture this was.
What lecture room?
Which lecture number this was.
Number 6.
Thank you.
You're welcome. So onset, again, since it's a foodborne infection, typically lasts longer, so about 70 hours before you get symptoms. Now this one is really interesting, and this is why the maple leaf outbreak was so dangerous, was if you are pregnant and exposed to Listeria, the likelihood of you either having a stillborn baby or a miscarried baby is extremely high. So just to give you an example, when you are a graduate student, if you're working with Listeria as part of your grad program, if you get pregnant, you have to stop your research. Because the risk of Listeria with infants and with fetuses is extremely detrimental. So here we talk about raw milk. Most raw milk is contaminated with a pathogenic microorganism. So you have to be very brave to actually drink raw milk. People that have been exposed to raw milk from very young ages produces antigens against these bugs, so they can actually have small amounts of the exposure and not get sick. But if you're someone who's never had raw milk and go and drink raw milk, chances of some kind of foodborne infection happening is very high. So how do we deal with this? Again, cooking. Most foodborne infections are prevented by the application of heat. Again, this had a huge consequence on Maple Leaf. They just settled the class action lawsuit for $27 million. And it was really interesting. I actually had a conversation with Chelsea about this, one of your TAs, because I'm not a microbiologist. And what was interesting about it was the meat wasn't necessarily the cause. So the meat was safe. It was the processing equipment which had been contaminated. So the slicing machine, they hadn't taken apart the slicing machine and done a clean of the deep insides of the machine, they say in the newspaper article. But what happened was they formed biofilm, which was then resistant to further cleanings. So this bacteria lived in the slicing machine, and every time a piece of meat went across that slicing machine, it contaminated it with Listeria. So it was extremely wide. 24 deaths in Canada were associated with that outbreak. So cold cuts. Cold cuts are a notoriously difficult food to ensure their shelf life. They're typically stored in an aerobic environment, and then meat is sliced off them gradually. So how can we control the growth? One thing that's being looked at the University of Guelph, which is really interesting, is a thing called phages. And phages are a microorganism that cause disease in bacteria. So it's like a virus to humans, but it's a virus for bugs. Now, when that phage gets exposed to a bacteria, it injects their DNA, their capsid, and this begins to replicate, and then we get phage control. So do you know in cold cuts where they give you those little pieces of kind of like parchment paper or wax paper that go in between each slice. What they've looked at doing is embedding phage into those surfaces. So when you put that phage on it, if there's Listeria, the phage will kill it. So then you don't have to worry as much, well, I shouldn't say worry as much, but you have another technological hurdle that you're putting in place so that there is a chance that it is contaminated. You eliminate that contamination after with the control of phages. And again, this is an example of a cell, and those black dots within represent all the different phages. And again, this cell will lyse, and those phages will be released just like a virus is in humans, and then it will go and infect more and more bacteria. So again, looking at that parchment paper, there's a lot of considerations in what you have to do to get that phage to be immobilized enough that it stays on that paper, that it's viable when it's exposed to the bugs, and that it can migrate from that paper to that food product and eventually to the microorganisms. So this becomes very useful, especially when we talk about thin slices of meat, where typically we're talking about surface contamination, surface growth of microorganisms. And Dr. Mansell Griffiths was a professor in the Department of Food Science. He retired a few years ago, but Dr. Keith Warner still works in this area. Now, so that's foodborne infections. Foodborne intoxications are when the bacteria again colonizes the epithelial cells, but now it's not the bug itself that causes disease, it's a metabolite of that bug that causes disease. So it's going to produce a compound that's toxic. So things like Clostridium botulinum, Staphylococcus aureus, or Clostridium perfringens. Now again, when we talk about foods, the risk associated with that food is dependent on the intrinsic factors of that food and the extrinsic factors of its environment. So in the case of botulism, and what food do you never give babies because of botulism? Anyone remember? Honey. Honey is not thermally processed. Why? Because it's mainly glucose. What happens to glucose when you heat it for a long time? It caramelizes, right? So when it caramelizes, the viscosity goes way up, the color gets more and more brown. You don't want that in honeys. So we don't thermally process most honey. But that packaging material we put honey in is aerobic, right? Oxygen can permeate in and out. So Clostridium won't grow in honey. It's there, it's present, but the environment restricts its growth because of the presence of oxygen. Now in infants and in young children, the pH of the stomach typically is higher than it is in adults. So our stomach acidifies down to a pH of 1.5. Clostridium will die in that environment. So if we consume honey and we get a little bit of Clostridium, it's going to be destroyed in our GI tract. In an infant that has a higher pH, that Clostridium can survive and actually sporulate, go from an aerobic environment to an anaerobic environment in the colon, and then all of a sudden it begins to produce the toxins and that baby gets very, very ill. When we talk about Clostridium botulinum, we are talking about the canned food, the aseptically packaged foods. So things that are in an oxygen impermeable packaging, so glass or metal. In North America, we are very rarely exposed to Clostridium. Typically, when people get sick from Clostridium, it's 1 or 2 people, and it's associated with home canning. I can't remember in my lifetime an industrial canner company like Campbell's has never been associated with an outbreak of Clostridium. With these types of intoxications, the onset can be much shorter, depending. So in Clostridium, if that toxin is formed in that can, you start to get the neurological effects very, very quickly. Onset time can drop to within hours. So typically between 12 and 36 hours. Now, again, we talked about botulism in honey. It can't grow in a pH below 4.3. pH of honey is above 4.3. If it's in an aerobic environment, we're fine. We have no growth. We don't can honey, or else we would have to thermally process it to get rid of the botulinum. The problem is, in infants, it can actually begin to colonize the GI tract because it goes into an anaerobic environment. Now, toxin-mediated infections mean that that toxin is what's causing disease. So Shigella and E. coli 0157H7, that's the one that was associated with the outbreak in Walkerton. This again, it is the toxins that cause a lot of lysis of cells, the epithelial cells. So typically in these, you see a lot of blood in your stool. As that infection gets worse and worse, more and more blood will begin to come out of the rectum. Now, when we talk about E. coli 0157, it is almost ubiquitously found in our food supply. In our cattle. So in cattle, about 70% of cattle have E. coli 0157 in their intestinal tract. 0157 does not cause disease in cattle. So they are a natural carrier for it. Now, the meat is not infected with the bug. It is all in the GI tract. So when we talk about slaughtering of animals, there's two processes in the slaughtering of animals that are extremely important. The first is the removal of hide. So that removal of hide has to happen very early on. It's one of the first things that happens. Again, there's going to be bugs on the surface of the skin. E. coli is going to be present. If we contaminate the surface of the skin, it can then be transferred onto the meat. The other process is evisceration. Evisceration is the removal of the intestines, the heart, all the organs. That's extremely challenging. If you have a spill, meaning if you're removing that intestinal tract and you have a rip and some of that intestinal fluid is put onto that carcass, you're supposed to cut away a large portion of that meat which has come into contact with it. So evisceration, they really, really want to make sure that all of those organs come out intact to prevent the surface contamination of meat. Again, meat itself is sterile typically. There's a few infections that can actually go into a living animal, becomes more relevant in pork and in chicken, but in beef, most bugs grow on the surface. So you can eat a raw steak, right? You can sear the surface, sear the bottom, all the bacteria is dead, you can then eat that middle meat pretty raw. When you commune that meat, when you break it up into small particles, make a hamburger patty, all of a sudden you can no longer assume that that surface is only contaminated, but that whole core can be. So you should never, like if you go to the United States and ask for a hamburger, they ask you how you want it cooked. The only option should be well done. People that eat beef tartare, especially if they're getting ground beef from, let's say, a grocery store, they're really taking a risk. Now, if you go to a slaughterhouse that you trust and you get a really nice quality steak and then you grind it at home, it's fairly safe. So if you go to a restaurant and eat tartare, so tartare is just ground beef with spices, right? So in restaurants, they won't buy ground beef from a grocery store. They will buy a whole steak, like a filet mignon. They will then remove the surfaces or sterilize the surfaces and then commune the meat. And with that, we're going to stop. And next week, we'll finish up the foodborne infections and talk about a little bit of processing.